First, I'd like to welcome everybody for joining us today as the kickoff to this molecular epidemiology training session. We're very excited to be able to offer this. My name is Justin Ball. I'm a professor here at the University of Georgia, and I lead a group on molecular epi of viral pathogens. The co-instructors for this course are Travis Glenn, who you'll be hearing from today, Elizabeth Otteson, who will be talking about some wastewater work. Antimicrobial resistance. She's popped on there. You can wave. And Aaron Lipp, who will be talking about wastewater surveillance. The goal of this training program is to provide you guys with greater context for the work that you're doing to help you with interpretation of your results if you're generating them. To hear about some of the cutting edge work that is being done by some colleagues and guest speakers that we're inviting in, as well as to hear about the ongoing work that is happening in the groups here at the University of Georgia, as well as some of the work that's being done by your colleagues in the Georgia Department of Public Health. The way that we've organized this training session, it will be 3 weeks per topic. We are starting with advances in genomic sequencing. We will follow that with viral molecular epidemiology. Then I believe it's wastewater epidemiology, and antimicrobial resistance. As we're progressing through this course, we hope to identify key topics that we will focus on in a follow-up in-person training session to get more hands-on work, especially for those individuals that would like to know how to analyze genomic data and understand some of the statistical approaches to making inferences that might be public health actionable. So this is the goal of this section. Don't know if Hope or anyone else from Georgia Department of Public Health is on, if they would like to say anything before we start. Quick chance. Nope. All right. Well, again, thank you so much for your enthusiasm. For the number of people that have signed up is very encouraging. We are truly committed to seeing genomic approaches being used to public health challenges and helping you guys develop and implement them in practice, especially as we're working on other questions within our labs. So we are very excited about this. Today, the first speaker is Travis Glenn. Travis Glenn has organized this session, and he is a professor here at the University of Georgia, leads the Institute of Bioinformatics and the Department of Environmental Health Sciences, and he has been a leader in developing genomic approaches and full genome sequencing of a number of different organisms and a number of important flags that have been planted in the progress towards our current state of molecular sequencing or genomic sequencing. So with that, I'm very happy to hand it over to Travis, and I'll let him introduce this session and get started with his talk. Thank you.
All right. Thanks very much, Justin. So I'm going to jump right in and see if we can catch us up. The goal is I'm going to try and finish by about 10 till 10. I think that's my goal. Somebody can tell me if that's not, if we're not on the same page. And then we'll do questions then. And you'll see I've got questions sprinkled throughout the talk that you can answer. All right, so again, just a little bit about me. So I'm a Midwestern farm kid. My background is sort of in biotechnology and ecology and evolution, and I've applied that in various ways. I've been in the Department of Environmental Health Science here at UGA for more than 15 years now, and sort of my emphasis is on environmental and genomic technologies. And so, as Justin said, I'm in EHS. EHS is part of the College of Public Health here at UGA, and I'm also part of the Institute of Informatics. All right, so here's your trivia questions, okay? And you can think about these, or if there's a chat function that you want to type them into, go ahead. Which came first, Sanger sequencing, capillary sequencing, yeah, which was used for capillary sequencing, or Star Wars? If you were here last summer, you should know the answer to that if you were in the training session last summer. Okay. Now the next question. Again, there's too many of us to have people just chime in, so I'm going to have you just write it down. All right, the next thing is, which came first, next generation sequencing or the first draft of the human genome sequence? I should have Jeopardy music playing in the background. Okay, final answer. All right, last question. Which came first, the first truly complete gapless telomere to telomere human genome sequence or the January 6th Capitol riot? Okay. You ready for the answers? Okay, so the answer, the first one's a little bit of a trick question because both of those were 1977. Star Wars came out in May. Sanger sequencing was published in December. Which came first, next gen sequencing or first draft of the human genome? Human genome by several years. The human genome first draft was announced in June of 2000. And the published in February of 2001. The first next-gen sequencers came on the market in 2005. The last trivia question was January 6th Capitol riot by more than a year. Okay, so what are the main objectives, right? So what we're going to try and do, what my lab does, is try and figure out how do we get DNA data, generally sequence data, to answer interesting questions and solve problems in public health. Okay, so again, you can write it in the chat function if you can easily find that, or you can just note this on a little piece of paper around you, whatever you have handy. And so what I want you to think about, I'm just going to give you, 30 seconds. Think about two things that you want to accomplish via DNA sequencing associated with your job, right? So what would you use DNA sequencing for? I need to have music in the background. Sorry, if we were on Zoom, I would be playing you music in the background, but I haven't figured out the music function on Teams yet. I could play it on my phone. Sorry. All right. So just a quick, what would you want to do? Okay. Then the next thing I want you, again, just take 15 seconds to think about, what do you want to learn about next-gen sequencing? Okay. I may or may not be able to cover that today. We'll see. But this is, before I get into it and distract you with a bunch of information, a blizzard of information, right? What do you want to know? Okay, and so if I don't get there, we can go back to that at the end. Okay, all right, so I'm gonna move forward. All right, so commonly in public health, the things that we want to do is look at a community of organisms and find the bad actor, right? Who's the pathogen? Who's the parasite? Who's the bad dude there, right? And then ideally we'd like to characterize that bad actor. What makes them bad? What are they doing relative to everybody else? And maybe we want to characterize that entire community because there's maybe multiple bad actors depending on the context or the role that they're playing at the time. Okay. So going from sort of an abstract thought to the, to the real world, what that means is if we're out sampling, either a healthcare setting or a non-healthcare setting, then how do we find those bad actors? All right, so traditionally what we're doing is go out, sample it, and now what we're going to do is, take that sample, extract the DNA, and try and find our target. And so there's lots of different ways that we might try and find that bad actor or characterize the community of bad actors. Okay, and those might range from, and hopefully you can see if I point on the screen, things like qPCR, or making a metagenomic library with number 3 there, or enriching that library for particular sets of bad actors that then we can sequence and identify and characterize better that way. Okay, and so today what I'm going to talk about mostly are the broad tools for sequencing. And then in the next 2 weeks, we'll talk about specific examples of how you would do things like take a metagenomic library and then enrich it for particular bad actors that you might be interested in. Okay, so what I want to do is, again, just sort of back up and make sure that everybody understands what's the difference between traditional Sanger capillary sequencing versus what we're really going to be focusing on with next-gen or so-called massively parallel sequencing. Okay, in Sanger sequencing, it's basically one tube, one locus, or one piece of DNA, one small piece of DNA for one individual. And it's physically separated from all other samples, individuals, and loci by a physical tube. Okay?
Get her,
And so one researcher would generally be able to do PCR to create many copies of that one locus of interest. And then you would do sequencing of that, one sample, which you can do in parallel on a 96-well plate. Okay, so you can do capillary sequencing in parallel, but it's not massively parallel, right? A 96-well plate is an array that lets you process a bunch of samples at the same time in the same way. Okay, so it is a parallel process. Okay, for Sanger sequencing, again, your reads are probably 600 base pairs. They're pretty good quality. And, bioinformatics is pretty important to help keep things organized, to process your reports and make sure that you know what's going on. Okay. In contrast, for massively parallel sequencing, you're going to get millions of sequences per physical tube. Okay. Oops. Hey. One researcher can do a few samples to several 96-well plates of samples per week. It depends on the platform. The read length can range from about 100 base pairs to over 100,000 base pairs. And that depends on the platform that you're using, the type of sequencer, what company manufactures that sequencer. Okay. Bioinformatics is crucial when you're dealing with that much data because you can't possibly look at it to QC it. It's just impossible to do that. So you have to use computers to automate that process. Okay. And so, like magic, next-gen sequencing makes previously unimaginable quantities of data a reality. And it makes it not only, is it possible to do, it makes it feasible by, within a normal research grant context, you can actually afford to get huge amounts of data. Okay. And even just 20 years ago, it would have been very difficult to imagine that we could get the levels and amounts of data and really now the quality of data that we're seeing from these new instruments. So what I'm going to do is sort of go through a variety of different instruments. We're going to emphasize the instruments that are most used by people, and in particular, folks who are in public health. So currently, there are 4 sequencing platforms, so 4 manufacturers of sequencers that have been around for years and years, and people understand those platforms. They're well used in different contexts. So I'm going to talk about these 4 different platforms. The oldest is Illumina, and so that's the industry leader. We'll spend the most time talking about Illumina. They have a bunch of different instruments, and I'll just try and give you an idea of the range and hopefully not sink into too many details on any one of them. Ion Torrent is the next oldest. Both of these are short read sequencers. And then Oxford Nanopore and PacBio are long read sequencers. And I'll try and distinguish, between those 2 platforms and the ways in which you would apply short read versus long read. Okay, so one way in which you might try and summarize the differences between these platforms is whether they read individual molecules directly, or whether they have to start with an individual molecule, then amplify it, so make millions, hundreds of thousands of copies, and then read that cluster of identical clonal molecules, or whether they can read that individual molecule by itself. Okay, Illumina and Ion Torrent start with a single molecule. They all start with a single molecule, but then they amplify that individual molecule that is spatially separated, but not separated by a tube. It's separated on a much, much smaller scale, and they amplify those individual molecules into clusters of molecules. Those are 2nd generation sequencers, with Sanger being the 1st generation. So these guys are 2nd generation, and then PacBio and Oxford Nanopore are 3rd generation sequencers, because they're reading individual molecules without amplifying them into a cluster. Okay. 2nd generation sequencers are short read sequencers. 3rd generation sequencers are long read sequencers. That's not always true, but it's definitely true in the way in which the market has developed. All right, so let's start with Ion Torrent. So Ion Torrent is a pyrosequencer. There have been many pyrosequencers. The first next-gen sequencer was 454. It is dead. Rest in peace, 2016. There are a variety of companies that used to offer pyrosequencers that are in the graveyard of time now. Okay, Thermo Fisher is the main one that is still alive that uses pyrosequencing. It is a mature technology. It is used largely for AmpliSeq and cancer screening. You can get a sequencer and the accessory equipment that comes with it, or you can get an entire system that goes from sample to report in a single day. The advantage of this is that the sequencing is actually quite quick. It's just a couple of hours to get the runs. The downside is that the scale isn't nearly as large as most of the other systems, especially the other short read sequencers like Illumina has a much larger scale. So the other thing that's sort of important to remember about this is the Ion Torrent was acquired by Thermo Fisher, and they've been doing a reasonably good job since then. But before then, Ion Torrent existed as a sole company that did not do a great job with customer support, and a lot of instruments were sold and created a lot of bad feelings about the platform, and that persists. So it could be useful in many contexts, but many people don't like it based on the history of the technology. Okay, Illumina is sort of the opposite, is that it was a company, a technology originally developed by a company called Solexa. Solexa was bought by Illumina, and then Illumina did a great job of customer support. It doesn't mean they're perfect, but it means they did way better than their competition. And that's how they ended up crushing, 454, is that they came out with new instruments and new ability, and they basically followed through with what they said they would in less than a year, beyond their promised date. Okay, so it wasn't that they weren't late ever, but they were late, by a couple of quarters, not by 4 or 5 years. So there are a lot of different sequencers from Illumina that range from the iSeq on the low end, which is about $20,000, and has one type of cartridge. It will give you exactly those kinds of results consistently. To the newest instrument is the NovaSeq X Plus, and I'll sort of show you that. It has a flow cell with 8 lanes, so it divides up a huge capacity for sequencing into 8 different addressable units that you would actually be able to use and purchase commercially. All right, and so to give you an idea of what's the range of instruments and what's the capacity that they have, I put a bunch of information onto one slide. Okay, I don't expect you to memorize this, but what I want to do is give you a feel for the range of instruments, which goes from sort of a couple of million reads per run to billions of reads per run. Okay, so it's relatively large scale. Again, these are short, so PE means paired end. 150 is the length of the read. So this notation up here is telling you which instruments have a maximum read length of 150 bases from one end of the molecule to 150 on the other end. The max for the entire platform is 300 by 300. Okay, and I've noted those for each of the different instruments. Again, you can refer back to these later. This is a resource for you to have not just while I'm talking, but into the future. Okay, so next gen is basically cheap per pound, but you have to buy the whole truckload. You can't just get a few reads at a time. You have to get millions or billions at a time. Okay, and so to give you an idea of how much we've saved in getting the sequences by moving to next gen, I've illustrated it in this way. So let's suppose that we have capillary sequences, right, on our Sanger sequencing. If you wanted to get 1 billion bases of data, that would cost you $1.4 million. But on the opposite side, the buy-in, how much does it cost me to get one read, is that it's only about $5. Okay, so the buy-in is cheap for one read, but if you want to get a truckload of it, it's very expensive. If we move to an Illumina MiSeq, it's about $1,000 instead of $1.4 million for the same amount of data. Now the buy-in is 100 times more expensive. Okay, that's the trade-off. I have to get millions of reads at a time. I can't get just one. Okay, and if I move up to the big NovaSeq X Plus, again, now I can get a much bigger savings on the dollars per billion bases of data, sequence data, but my truck is getting even bigger, right? So I've moved from a pickup truck load to a dump truck load. Okay, and this is per lane. right? Remember, there's 8 lanes on one of those flow cells. So commercially, you can buy by the lane, and your run is separate from everybody else's. And the person who has that sequencer is putting together different people's samples to go on that same run. Okay. Now, if you have this instrument in your own lab, you can do the same thing, but ultimately you have to pull the trigger on and run an entire flow cell. And those big flow cells are on order of $15,000. Okay, so relatively expensive. You know, it's a big truck. Okay, so now what if we sort of compared the cost of getting 1 million reads on any of these different Illumina sequencers? So public health labs almost all have Illumina MiSeqs. And the cost of getting 1 million reads, if it's at a standard length of PE 150, so I'm going to keep that the same. If you go longer than that, it will be more expensive. Okay. And I've done it. I've got 2 numbers here. One number is the commercial cost. If I go somewhere and send my samples and I ask them to run at a commercial facility and send me back the data, what does it cost? And then in parentheses is I own my own instrument. The technician is already paid for. I'm just paying for the cost of the run, the reagents. I'm not accounting for the annual service contract or anything like that. Okay, so again, if I jump down here to MiSeq, that range can be from $90 on the biggest flow cell to more than $400 on the smallest MiSeq Nano flow cells. Okay, the ones with the lowest capacity. So it can cost, you know, $100 or more per million reads. If I go down to these big instruments, that cost can be down to $1 or $2. Okay, so the savings is substantial by moving on to these higher throughput instruments. If you have a project that doesn't need a ton of reads, then a MiSeq can make sense because it lets you process samples in a quick way. Okay, but if you have samples where you're trying to get millions, many millions of reads, tens of millions, hundreds of millions of reads, then you want to be down here in NovaSeq land, paying only $1 per million reads. Okay, so overall, what are the strengths of Illumina? Illumina has had more than 90% of the next-gen sequencing market for more than a decade. And the reason for that is that it's cost-effective relative to the competition. It has good reliability. It has had excellent customer service. I mean, it's one of the few companies that's like, I look forward to asking their, customer service for help because they're really nice and they actually will look for stuff and they'll get the answers back to me, right? It's an unusually pleasant experience. The final thing is that their applications are really good, and they were smart and they made most of their system open so that researchers could understand how the system worked. They could tweak it in their own ways, plug it into, the parts that Illumina was selling, and everybody would benefit at the end. Okay, so that is a model that other companies have adopted now, and I would say almost all of them have adopted that basic position, and it really let Illumina flourish. Okay, they also have many instruments and kits. The sequencing technology has a good bit of flexibility and openness, right? So again, people can innovate. It is basically the industry standard. If somebody, is contemplating a next-gen sequencing project or project that needs next-gen sequencing, Illumina should be your default. And then you compare all the things, other possibilities relative to that default. Because Illumina isn't always the best answer, but it's frequently, it's rarely a bad answer, if that makes sense. Okay, so if you're risk averse, it's it's sort of a good default position to be in. All right, so what are the competition? So Pacific Biosciences. So Pacific Biosciences is doing real-time single molecule sequencing. Their newest instrument, which came out last summer, is the Revio. It runs up to 4 flow cells independently / simultaneously, which means you can run 4 at a time, but you can load them independently. So you can load 1, start it running, then load number 2, start it running, blah, blah, blah. Okay, each run is approximately a day, plus or minus a few hours. The flow cells have 25 million wells in them. The occupancy rate is less than 50%, so, and usually it's maybe a third to a quarter is still a pretty good run. The molecules that you're sequencing are quite long. They're on order of 1 to 25 kb, and so 1 kb is sort of in the amplicon range, and 25 kb is the standard, genomic library preps. They use an approach called hi-fi sequencing read, which means it's a circular consensus. So they take a molecule, the adapters on the ends are hairpins, so it makes a circle so that you can read round and round the same template molecule. Okay, and that results in a high quality read. It is the current default for most de novo sequencing of genomes. So if you're sequencing an organism for the first time, or one individual you're particularly interested in, and you want to know, not comparing it to what's known, just from the data I get, how do I assemble the genome sequence? PacBio is currently the best way to do that. Okay, it's not always the best, but it's now the default position, the default platform, if that is the thing you're targeting for your project. All right, so again, Revio was introduced just this past year. It's been on the market for less than a year. Its cost is about 3 times the cost of Illumina, but that cost premium is worth it for some applications. And again, maybe the most commonly used application is de novo sequencing of individuals that you're really interested in. All right, what's the major competitor to PacBio? It's Oxford Nanopore, which is abbreviated ONT. So Oxford Nanopore determines a DNA sequence without synthesizing a new strand. All of the other technologies are using a DNA polymerase in one way or another to create the DNA sequence that you see. Oxford Nanopore feeds a DNA or RNA strand through a nanopore, and then it looks at the electrical signal that's generated during that. That signal changes depending upon the sequence that's being read through it. Okay, so they are, looking at intact whole molecules, 400 nucleotides per second. Okay, so it reads the DNA or RNA very, very quickly, but the diffusion process of getting the DNA or RNA to the nanopore is slow. So typical runs are oftentimes 3 days long, okay, because you're waiting for that diffusion process to end. Oxford Nanopore has a range of instruments which range, sort of in size from a giant king-size Snickers bar up to sort of a small printer. And so these have various portability. There's a bunch of different options that can do all sorts of amazing things. Okay, so Oxford Nanopore sequencers are particularly low cost to purchase the instruments, and the data within the past 2 to 3 years have really crossed a quality threshold that makes them usable in a wide variety of ways. Prior to the version 9 nanopores, you kind of had to stand on your head to use the data. Since version 9, and now we're on version 10 of the nanopores, the data are significantly better. One of the major things is that they have 2 read points within those new nanopores. So you're actually getting 2, so sort of similar to circular consensus sequencing with a minimum and maximum of 2. Okay, and that has helped them a lot. The sequencing technology has great potential. Again, it's relatively open, so Oxford Nanopore allows their users to innovate a lot. Okay, and it's been clear since, you know, the very first introduction of the technology that it was going to be, you know, it had the potential to do amazing things. Now, it has been more than 10 years since the platform was introduced, but people have maintained that enthusiasm, you know, in a way that the Ion Torrent could not. Okay. All right, so what are some of the contrasts between PacBio versus Oxford Nanopore? All right, so PacBio is a more mature, stable platform. It's been around longer. They don't oversell what the instrument or software will do. They're very good about telling you this is what the instrument does. Now, again, they haven't always done that. They did that after they brought in a CEO about 5 years ago who's like, no, we can't just oversell promises and wishes. Okay, so it's not that PacBio always did this. The first 6 or 8 years of the company, they were making crazy promises that they could not deliver on. Okay, but since then, they've really turned things around. The bioinformatics is easier. The downside is it's really only one instrument, the Revio, one long read instrument that's around, and they only have, you know, one flow cell option. So everything has to go through that constraint point. Oxford Nanopore is still evolving rapidly, which is sort of good and bad. It's good because it continuously gets significantly better, but it also means that you have to change stuff relatively frequently, right? They still persistently oversell theoretically what their instrument could do, which no instrument has ever done ever. So I don't get why they still do that, but they do. Okay, the bioinformatics is harder. Okay, but more people are using it, so it gets easier as time goes on. There are many instruments. The cost of those instruments is lower. The cost of the reagents is lower. So your ability to buy into it is much better. Okay, so what are some differences between the PacBio reads and the reads you would get out of the highest throughput Oxford Nanopore instrument, the PromethION? Okay, PacBio reads have much higher quality. PromethIONs are lower quality. They are coming out with 2D reads, which is what the T2T, the telomere to telomere human genome draft used. And so if you have access, early access to that, that technology and software, then that gets it closer, significantly closer. But you need a lot of DNA. I mean, hundreds of micrograms of DNA to do that. So they're still working on that. The yields, you know, for an assembly for a PacBio, again, that's just put it in, turn the crank, you're going to get a good assembly. With ONT, it's you got to know what you're doing and do some fancy trickeration sometimes. So the bioinformatics is easier on PacBio side. It does require more DNA for the library preps for PacBio. And again, it's higher cost to get into. All right, so what are some of the competitors that you might hear about? So you might hear about Singular Genomics. I don't think in public health that has any potential forward, so I'm just going to skip through that one quickly. Similarly, for Ultima Genomics, it is the other pyrosequencing platform that's around, but it only has one sequencer. It's at a huge scale. The only application really is full human genome sequencing or something similar to that. The cost per human genome sequence is quite low. So you will find these in some of the biggest sequencing centers, but you're not likely to see this really used extensively in public health. Will they come out with more instruments? Maybe. We'll see. All right, now one you're more likely to see are sequencers from MGI Tech. So MGI Tech essentially has a platform that uses a different way to come out with Illumina-like results. Okay, they have a range of instruments that depend on patents that they were fighting with Illumina for. And a little over a year ago, year and a half ago, Illumina ended up paying MGI Tech $400 million. And then Illumina's patents expired. And between those things now, MGI Tech can offer their full range of instruments in the US. This is a company that was previously, spun out of BGI, which was the biggest genome sequencing center in China. The MGI Tech instruments are lower cost and the reagents are lower cost versus Illumina. So it is a viable commercial alternative. The difficulty probably for most public health agencies is that because of this history with China, even though there is a US subsidiary, components are made and constructed in China, which will create some significant hurdles to purchasing it using federal dollars. The last new sequencer that I want to talk about is a US company, Element Biosystems. They have an instrument called the Aviti. Again, the Aviti is a totally different way of producing Illumina-like results. One of the best things about it is that you can literally take Illumina libraries and convert them into libraries for the Aviti. So you can take all of those things that you've spent years and years validating, getting to run on Illumina, and then you can take the pools, and instead of running that pool on an Illumina, you can do a quick library conversion and then run it on Aviti. And the reason that you might want to do that is that although the instrument is sort of similar to a NextSeq 2000, again, it's cheaper to purchase the instrument and it's cheaper per run, and you get more data from the biggest flow cells. So they sort of offer a flow cell that's one step bigger than the NextSeq 2000. The NextSeq 2000 ends at a P3 flow cell, and so if you imagine there was one step bigger, that's what a Aviti opens up. And so there are some big sequencing centers that have these instruments available, and there are some medium-sized labs that are acquiring these and using them instead of NextSeq 2000s. All right, so here's your quiz. We're going to go back and do the quick review in the next 2 minutes, and then sort of try and wrap up here. So what is the biggest, the most commonly used platform? Everyone should be thinking Illumina. Okay. How does PacBio achieve high quality sequence reads? What was it they did? Oh yeah, they went round and round on the same template molecule so that they could end up reading the same thing. So circular consensus reads make those hi-fi reads. Okay. How can you identify or verify that your reads are high quality? Okay, there's this thing called Q-scores. I'm not going to go into this, but the gist is the quality score, which is a Phred or Phred-like quality score, is a log scale where the first number in the tens place tells you the percentage of errors in your sequence reads, okay, for any one position, okay? So 1 is a 10% error rate, 2 is a 1%, 3 is 0.1, 4 is 0.01% error, okay? So a Q40 is very, very good. A Q10 is not so good, okay? So what Q number indicates high quality for you? Okay, and again, this is one of those trick questions. It depends on the application. Some applications, you don't need the quality score to be super high. Other applications, you need the quality score to be very good. Okay, and there's a trade-off. And I'm going to illustrate this to you with one last example. Okay, so let's suppose that we have an E. coli isolate and we want to sequence the genome and assemble it. Okay, and so this is an exercise that we did with a workshop a couple of summers ago, and we had 3 data sets. One was an Illumina MiSeq PE300s. Okay, and so when we did that, the genome sequence was quite good. There were very few errors. It was low cost to obtain, but it had hundreds of different pieces, so it didn't assemble it into one chromosome like it should have. The other side of that was that we took Oxford Nanopore data from MinION. And again, this was like version 8 or 9 data, early version 9 data. And this was, so it assembled, because the reads are long, into one contig, right? One chromosome-sized piece. And the cost was higher, but not too much higher. But the problem is it contained hundreds of errors, as the error rate, at least on data obtained from that time, wasn't as good as it needed to be. And the third data set that we had was PacBio HiFi. And so this was at a time when the HiFi data were more expensive because the Revio wasn't out yet, but it had very few errors. It assembled into one piece, but the cost was high. Okay, and now with the Revio, the cost is lower, but it's still not as low as these other 2 alternatives. Okay, so these are the trade-offs that you need to think about when you're contemplating which next-gen platform are you going to use. Okay, the last thing, preview for next week's talk, right? The basic gist is, now next week, what we're going to be talking about is how do you find those pathogens in, a complex sample. So we're looking for that bad actor, that needle in a haystack. And the short answer is, we're going to use magnets. Okay, all right, so you'll hear all about enrichment techniques next week. And so there are many, many people I'd like to thank. I'm not going to take time to do it, but obviously my work has benefited from collaborating with a huge range of people, including Justin. Thank you, Justin, for the opportunity to speak today. I am going to stop sharing so that I can see if people have questions.